Right, this is going to be a review of lesson 3 on meiosis, which is on traits and probability. We'll be covering Punnett squares, dihybrid crosses, and pedigrees. So this will help you both on your notes packet as well as your Punnett square and dihybrid cross worksheets. So we use a Punnett square to model a genetic cross. Just like we model a solar system or the galaxy, we model what would happen if two individuals with particular traits had offspring. So this was developed by a guy named R.C. Punnett, and this is to help predict what genes will be passed on. So we want to remember our vocabulary here. A genotype indicates the alleles that an organism carries. For instance, if somebody is dominant for a trait and recessive on the other allele, that means they're a heterozygous genotype. And maybe this person also has a heterozygous genotype. And this could be for anything. So let's say it's for flower color, right? So we always want to have a key here and say like capital A, the dominant trait is going to equal blue flowers. And the lowercase a here is going to equal yellow flowers. All right, so that would be our genotype, and then these would be our phenotype. So when we do one of these crosses, we want to take the large letter here, whatever letter is at the top here, whatever allele is at the top, and drag it down. Same with this one here. And these we want to take over and basically fill in every box. So this letter is coming here and here, and this one is going here, there. Now, it does not matter if the capital letter is first and the lowercase is second. This and this are the same thing. We just tend to write the lowercase letter second because it looks nicer. So this is the basic practice using a Punnett square. And this is to calculate probabilities. So when we look at this, we can see that there is a 1/4 chance that the offspring is going to be homozygous dominant, or in our case, have blue flowers. There is a 50% chance that they're going to be heterozygous, which also means they have blue flowers, and a 25% chance that they are homozygous recessive, meaning they would have yellow flowers. So we could write this in two different ways. We could say that 1/4 or 25% are going to be homozygous dominant. Sorry, guys, they're right there. All right, and we could talk about the probabilities of their genotype, which would be the homozygous dominant. We would do that for all of them. So we'd say 1/4, 50, and then 1/4. We could also say that 75% would be blue and 25% would be yellow. And this would be talking about the phenotypes. Basically, in that example, we are saying that all of these, this 75%, are all showing up blue because they all have at least one dominant trait, whereas this 25% are showing up yellow because they have two recessive traits. So really the best way to understand Punnett squares is to practice them. So we're going to look at 3 types of crosses. The first is going to be a, writing here, wrong thing, going to be a homozygous with a homozygous cross. So this is basically be saying if we have capital A, little a, these would come across like this. And all individuals would be heterozygous. Okay. We can also look at a heterozygous cross long words to write. And this would be that both parents are heterozygous for the trait, so they both have one recessive allele and one dominant allele, which basically shows that we have big A's come across, little a's come across, big A comes down, and little a comes down, which then we get the same pattern that we saw before, and we see that 25% are homozygous dominant, 25% are homozygous recessive, and 50% are heterozygous. Lastly, we could have a heterozygous homozygous cross, which in this case, I forgot a letter there, but that's okay. In this case, we have one heterozygous parent and one homozygous parent. And so this would change depending on if the parent was homozygous dominant or homozygous recessive. But either way, you can figure out the probabilities by just filling in the little box. So we can see here 50% of the individuals will be homozygous recessive, meaning they'll show the recessive trait, and 50% will be heterozygous, meaning they'll be carriers for the recessive trait, but they will be showing the dominant phenotype. So again, we can practice some more here. So I can give you any combination of letters here we can look at. And they could be, and you can figure them out. So for instance, look here, and you can see I'm just bringing the letters down and over as I come across. So this letter is coming here and here. This letter is coming here and here. And so this becomes a very simple process. And then after that, it's just kind of doing math, and it's saying, okay, how many of these are homozygous dominant? How many of them are homozygous recessive? How many are heterozygous? And then you want to look at phenotypes. How many of them have at least one dominant allele, meaning showing the dominant phenotype? How many have no dominant alleles, which is showing the recessive trait? And so you'll be practicing this a lot on your worksheet. So if you really understand this, you can be going ahead and working ahead on those problems. Right? So it's a pretty simple process. If you have any issues with it, it's mainly going to be probably with vocabulary, not filling in the boxes, and we can go over that here in class. So now we're going to make it a little bit more complicated. Now we have dihybrid crosses, and this is basically going to cross two traits at the same time. And so we're looking at saying, okay, if we have a tall green plant with a tall green plant, so they're both tall green, but they have different genotypes here, what would happen? So first what we have to do is we have to figure out all the possible gene combinations they can get from each parent. So we're going to do this in two different colors here. And look, because that just makes it a little bit easier. So we're going to say we have the tall green plant here. That'll say that's the mom plant. And we'll say the dad plant is here in blue. So let's start with the mom plant. We can see her genotype. She is homozygous dominant for being tall and heterozygous for being green. So because these traits sort independently, she could show any of two or four different genotypes. So to show you this, she could have this tall genotype and that green genotype. So she could have a capital T and a capital G. So we took this one here and that one here. She could also have this tall allele with the capital G. So that's another capital T and another capital G. So she could have this one and this one, or so she could have this one and this one, or this one and this one. Now there's two more options. She could give this allele here again, so this capital T again, and also give the little g. So that would be capital T, little g, or she could give this capital T and little g. All right, so those are the four possible genotypes she could give. So we're going to write those up here along the top. Right, so that's for mom. For dad, we're going to do the same thing. His genes, he could have a dominant allele or a recessive allele for tall, or a dominant allele and recessive allele for green. So for Dad, he could give these two, which would be capital T, capital G. He could give these two, which would be little t, capital G. He could also give the capital T and the little g, so the dominant trait and the recessive trait, or the recessive trait and the recessive trait. So these are his 4 traits he could give. So we're basically distributing these combinations of alleles and trying to figure out how many possible combinations are there. So this would be capital T, capital G, little t, capital G, dominant for tall, recessive for green, recessive for tall, and recessive for green. So now we do basically the same thing. We bring these variables, or we bring these alleles down and over into the boxes. So we'll start out with Mom here and say, okay, Mom is going to bring a capital T to all of these boxes and a capital G to all of these boxes. Notice I'm leaving space for the data alleles. And so we can actually do the data alleles now. He's going to bring a capital T over and a capital G over to all of these boxes. And so we could do the same thing here. Notice I'm just copying whatever letters are over here into the box. And so we get a view of the potential offspring. So instead of having 4 potential outcomes, we now have 8 potential outcomes, or 16 potential outcomes. So we're going to go ahead and continue to do this, so kind of follow along. This is actually from your dihybrid cross worksheet. It's number 1 on there. So you can look at this with me. And you can see, you can bring them, you can do one little box at a time, or you can just bring them all the way down, like I like to do, and fill them all in and then count. Okay, and so then we'll go to Dad here, and we know he has little t's across here and big G's. And big T and a little g. And finally, 2 recessive traits here. So what this shows us, and I'm going to erase our FOIL methods over here, but don't forget how to distribute these variables. What this actually shows us is that some of our individuals are going to be tall and green. Some of our individuals could be tall and yellow. Let's say it's not green, so it's going to be yellow. And there could be the potential, I forgot a few here, there could be the potential that some plants are short if they have 2 little t's. But we can see here, because Mom has no little t's, none of the offspring are going to have 2 little t's. So we'll look at that. So what we want to know is how many individuals are both tall and green. So means that this one is tall and green. So that's 1, 2, 3. Even though this has a little g, there's a big G there. 4. This one is tall and green, tall and green, tall and green, tall and green. So that's 8, 9, 10, 11, and 12. So we have 12 individuals that would be tall and green. They have at least 1 dominant green trait and at least 1 dominant tall trait. We then have tall and yellow. We have 4 individuals. These 4 have 2 little g's, which means they're going to be yellow, but they're still tall. So we have 4, and that of course equals 16, so that's everybody. When you're doing your questions here, sometimes how many would be short and green? And that would of course be 0, and short And yellow would, of course, also be 0. And so these are your phenotypic ratios. And so this is a really useful for when we're looking at more than one trait all at the same time. Again, this takes some practice, so you want to be practicing those on your worksheet. So this is great for when we're looking at, what if somebody with blue eyes had a kid with somebody with brown eyes, and we can do these little Punnett squares and figure out, or hitchhiker's thumbs, or cleft chins. We can figure out one individual pair. But we also want to look at how traits are passed within a whole family. Because you can't go back and ask your great-great-great-great-great-great-grandmother if she had a hitchhiker's thumb. Chances are that was not recorded anywhere. And it really doesn't matter. But when we're starting to talk about things like family history of hemophilia, for instance, then it starts to matter a little bit more. So we want to use conclusions to determine patterns about how genes are traced within families. What we use is something called a pedigree. So a pedigree is a family tree, and they have a basic code. Squares are males, circles are females. Unaffected people who do not have the trait, let's say it's hemophilia, which means your blood does not clot. So let's say that people who do not have the trait, they're not filled in. People who do have the trait are filled in. If there's a line between them, that means they mated or had kids together. So you can see here you had an affected female, so a female with hemophilia, and a male with hemophilia over here. This person married this guy. They had 4 kids, and none of them had the trait. These 2 people married, their one child did not have the trait. Their 2 children marry, one of their children has the trait. So we can look at how the trait was passed on. Chances are this person carried the recessive allele, and this person carried the recessive allele, meaning one of their children got the doubly recessive, which gives them the affected trait. Because remember, we're looking at dominant and recessive here. So anybody that's unaffected has at least one dominant allele. And anybody that is, so any unaffected people have one dominant allele. Any affected individuals would have this phenotype. So we can guess the genotype based on if they were affected or not. You also have, a little bit more complicated ones here. And so you can look at the pattern and say, okay, we know this is a recessive trait because it skipped a generation. Whereas this one is not skipping a generation, but it's only showing up in males. So one female gave the trait to all of her male offspring, but not her female offspring. And then the female offspring was still able to pass that trait on to her male offspring, and the male offspring only passed it on to his male grandson that came from a female here. So what this tells us is that this was an X-linked trait. Because remember, you only get the X chromosome from your mom if you're male. So all of these males would have got the X chromosome from their mom, which means it had the trait on there. But the daughter, she had 2 X chromosomes, so she got one normal chromosome from her father. But because she had the 1 X chromosome that was a carrier, she still passed it on to her sons. This young man was able to, he had the X chromosome, but his sons got their X chromosome from their unaffected mom here. So they got X chromosomes from somebody else. However, his daughter got that affected X chromosome from the mom, but she also got an unaffected one from her mom, so from the grandmother there. And then that trait's down here. Basically, what you need to know about pedigrees. If mostly males have the trait, it is probably an X-linked trait. Whereas if you have a range of both males and females having the trait, then it is probably an autosomal trait. So, reading these pedigrees, reading the paragraphs that go with them, is going to be important when answering this question.